So, welcome back from the lunch. We'll start the session now. So my name is Rikard Holmdahl, and I'm a member of the prize committee. I have the pleasure to introduce Professor David Nemazee here. And for this prize, as you know, there is shared between two laureates, Chris Goodnow and David, and they basically worked out the B-cell tolerance that they got the prize from very independently. They didn't know of each other's work until the end here when they published papers, as I learned. So it's really parallel work in different ways, which is amazing. It indicates also how science works, that the technology come to some point, and there are some smart guys that figure out how to do it, how to use it, and break the dogma. So David was born in Iran, actually, and moved to US when he was 3 years old, correct? And then made a PhD at Harvard. After that, he had a good idea to move to the Basel Institute in Europe, which is very has contributed a lot to immunology, especially to B-cell immunology. Basel Institute, for example, has produced Susumu for having the VDG recombination findings. So, and they also were early on to establish transgenic technology. So I think it was a good environment. And this is the environment where the first mice was done for making the B-cell tolerance data that was published. David later moved on to Denver in US, and after that being a professor at La Jolla in California. The work was followed with many more papers. So what David was doing was with the same question posing that Chris Goodnow had, how B-cell tolerance could be addressed in different ways. And here they had a mice they made a mice with an antibody that was binding to MSC class 1 antigen. It's a common MSC antigen basically expressed on all cells. As you know, this is a high polymorphism of class 1. So certain mouse strains have a certain allele, other mouse strains has another allele. So they had an antibody against the K allele, and they expressed that in the mouse with the D allele. So it's normal selection. It's not a self-reactivity. But then they crossed it to a mouse with the same allele, with the K. Then you get deletion. And MSC class 1 is membrane-bound antigen and high affinity. So this was the first example how B-cell tolerance could be induced with deletion in a conclusive way. And he then continued to look at why you get that deletion. And they found a new mechanism called receptor editing in parallel with another group led by Martin Weigert. And in this was completely new and very surprising, in fact. So the question was this, that if you just delete B cells and have other to select that is not self-reactive, you will waste a lot of B cells, basically. And to not wasting this, you could change the receptor another time, which was against the dogma at the time here. And that's called receptor editing. So you could recombine it again and find another receptor ongoing that was more useful. So, and we know now that many B cells in the repertoire are actually receptor edited. So with that, I invite David to come stage and give a lecture for us. Welcome.
Thank you. Well, thank you so much for the kind introduction, and thank you to the Academy and the Crafoords for this wonderful prize. It's quite an honor, and Yeah, I'll be enjoying it for many years to come, I'm sure. Yeah, so thank you again. I really appreciate it. So I just want to briefly talk about B-cell tolerance in the first third or so of the talk, and then talk about other things we've been doing. Basically, you know everything about, the people in this room know everything about B cells and how they get expanded when they see an antigen, and how they can class switch and be somatically mutated. But I wanted to point out or something that's fundamental to the immune system that our work sort of illuminated, I thought. So as you recall, the heavy chain genes are composed of 3 gene segments that get assembled together by the VDJ recombinase process to give you an expressed heavy chain. Whereas the light chain genes, kappa and lambda, have V's and J's, but no D elements. And I remember when studying this, it seemed odd and arbitrary, and all the textbooks said that you do this to increase diversity, but all of the diversity really is in the heavy chain. You make millions of different kinds of heavy chains and only thousands of different kinds of light chains. So there's an asymmetry there that when you're learning this stuff, you just accept. But it turned out, I think, to be fundamental. What I want to talk about first is the quality control problem in B-cell development. So the random rearrangement process that generates the receptors causes a lot of problems, actually. You have to avoid expression of more than one specificity, and therefore you want to rearrange one functional heavy chain and one functional light chain, and not a lot more than that. And to make things worse, on the light chain locus, you have 2 different kinds, kappa and lambda. And this makes it all more difficult. And in addition, most of the rearrangements are out of frame. So you rearrange elements together that will be non-productive most of the time. And on top of that, you have the problem of self-reactivity. So the rearrangement process is so random, of course, you generate self-reactive cells. So I think our work somewhat illuminated some of these issues. And so the other asymmetry in B-cell development is the order in which these gene segments are rearranged. You first rearrange the heavy chain genes, and that's done separately from the time period when you rearrange light chain genes. And to test that heavy chain for functionality, you assemble it with the pre-B-cell surrogate light chain components. And this transmits a signal to allow the B-cell to advance in development. And then the surrogate light chain proteins disappear, and rearrangement is redirected to the light chain loci. And when you could make a good light chain, that is detected by the assembly of the whole B-cell receptor on the surface. And that, in a sense, is a positive selection signal for development. So by this way of thinking, light chain expression promotes a recombination stop. Once you made the heavy light pair, you don't need to recombine anymore. This is what I was mentioning before, the fact that heavy chains have D elements, many of them, and these VD and J elements are rearranged more or less randomly and independently assemble. You make many combinations of these, and there's junctional diversity at the junctions, which leads to millions of heavy chains being generated. Whereas the light chain loci have just V's and J's, And there aren't that many of them, and so you don't make many combinations. And they can't rearrange with each other. In other words, V lambda can't rearrange to J kappa. So these light chain loci don't really contribute that much to diversity. They also have usually, they only have one junction, so there's less junctional diversity. So quite an asymmetry here in diversity. So our work actually was, which started back in Basel, was made possible by the groundbreaking work of Brinster and Ursula Storb and others who made the first transgenic mice with immunoglobulin and could show to various extents that if you express a pre-rearranged immunoglobulin, that you could suppress to some extent endogenous rearrangement. And it's at that time it was presumably through the sensing mechanism for functional chains. When this and one key experiment was from Rusconi and Kohler, who made a mouse that co-expressed heavy analyte rearranged genes, and they got a mouse where where many of the B cells made an antibody to that antigen, which was an anti-hapten antibody. So that suggested that if you provide the system with pre-rearranged transgenes, you would tend to suppress the endogenous rearrangements and get a relatively monoclonal B-cell mouse. And so we thought we would try this with an autoantibody. And the key, my contribution was making the constructs from an antibody that was of interest, which was an anti-MHC alloantibody derived from a BALB-C mouse that saw many different MHC types from other mouse strains. And the key collaborator for me was Kurt Bürki, who actually was in a pharmaceutical company at the time, Sando, in Basel. And he was a key collaborator. And it was Kirsten Fischer-Lindahl who introduced me to Kurt, who told me that he was the guy to talk to. And so we were able to do this experiment, make these mice. And we made a couple different versions. The first version was just an IgM version. So we co-injected heavy and light chain genes for an IgM anti-MHC. And then later, with the help of Zlatko Dembic, we extended the heavy chain to include IgD here. And we were able to make these transgenic mice to study B-cell tolerance. And the key difference was these mice were leakier than these for reasons which still aren't all that clear. But the latter mice, it was very clearly a monoclonal B-cell mouse where if the antigen, the cognate antigen wasn't present, virtually all the B cells in this mouse carried the 383 immunoglobulin receptor. So this is on the H2D background. No autoantigen is present, and you get a nice system here. And we tracked the clone using an anti-idiotype antibody I made. So this isn't the order in which the studies were actually done, but this makes it easiest to understand. So if you place the autoantigen, so this antigen here, K of B, for this particular antibody is a cross reaction. It binds very weakly to this antigen, but it's sufficient for tolerance induction. So this is a transgenic mouse that we obtained from Jacques Miller's group in Australia that expressed K of B on the liver hepatocytes. And we bred our mouse to this one to see what would happen. So here the B cells are generated, they wander out into the periphery and will bump into the autoantigen. And the results were amazingly clear. So if you look in the bone marrow, spleen, and lymph node of the mouse lacking the autoantigen, all the B cells express the clonotype, the idiotype of the transgene. And then if you look then in the double transgenic, where the cognate antigen is present in the liver, there's a strong reduction in the frequency of B cells, and in the spleen and in the lymph node, you basically have almost no B cells left. So this was an important experiment for us, as it showed that the B cells were generated in the bone marrow normally. They even upregulated IgD, which was thought to have some role in tolerance, and yet the B cells, which were made in huge numbers constantly, were readily tolerized in this system. So this fit with the clonal selection hypothesis that autoreactive cells could just be eliminated. So you weed out the autoreactive ones in a very efficient way, very quickly, and over a long period of time. The striking thing is that we obtained different results when the antigen was present everywhere on the whole body. And that's a simpler experiment technically, because there are many MHC mouse strains of different MHC types that would react with this B cell. And this is one example of the results we got with the IgM-only mouse, where we could see that in the absence of the autoantigen, we have lots of B cells with this clonotype. But in the presence of the autoantigen in the spleen, we don't see the autoreactive cells at all. And we always saw a population of cells that lost the transgenic specificity or never had it and expressed IgM. If you look in the bone marrow of the same mouse, we saw sort of downregulation of the B cell receptor, which we interpreted at the time as being deletion during B cell development here. When the autoantigen isn't present, you see a lot higher density there. Anyway, so what was really made us think was that, when we analyzed those few idiotype negative cells that persisted in these mice, they expressed the transgene heavy chain, which we could show with allo antibodies, but endogenous light chains. So the clearest example was with lambda light chains. Because our transgene was kappa, we knew these had to be coming from endogenous genes. And we worked out that in the absence of autoantigen, the expression of recombinase was low in the bone marrow, and it was upregulated when antigen was present. And so our model, and we also saw more variant cells. So if you look for lambda, when antigen is present in the whole body, there's much more numerous than when antigen is present just in the liver. So this suggested that the appearance of the lambda positive cells was induced and not the result of selection of pre-existing variants. So our model was that if you have an innocuous receptor and you're developing in the bone marrow, that turns recombinase off. But when antigen is present, then recombinase stays on. You keep rearranging, and you can change the receptor, specifically the light chain part of the receptor, okay, which is shown here. And once you can get rid of that autoreactive light chain, you can frequently fix the specificity to something more appropriate. So the model then came like this. You could potentially save a lot of cells by editing them to a different specificity. So this was not something we were looking for, but it is a new way of modifying autoreactivity. You can delete the receptor without deleting the cell. And I found it very beautiful, the fact that it helped me to understand the asymmetry in the light and heavy chain genes. So there's this, the recombination signals that put the gene segments together have a certain polarity. So you have a 23 base pair spacer and a 12 base pair spacer recombination signal that get put together. So on the heavy chain, when those are all assembled, you have upstream V elements and downstream J's that can't recombine together. Whereas on the light chain locus, there's really nothing to prevent secondary rearrangement. So when a V rearranges to a J, you make a functional exon that can be deleted by subsequent rearrangement like this. So this is the way it would work in a physiological locus. The fact that we saw it in the transgenic was just partly luck and partly the fact that we got rearrangements within the transgene unit. And then there's, yeah, well, let me skip through this now. So our model is basically that in the bone marrow, something different happens than in the periphery. In the bone marrow, when antigen is present, you have a pathway to edit. And if that's blocked in some way, as you can show if you make these transgenic B cells RAG deficient, so they can't rearrange, the cells die, or there are other situations where you can demonstrate that. But what's much more efficient is to edit away the receptor than to kill off the cell. And then at a certain point when the B cells mature, they lose the ability to be edited by self-tolerance. And that's why in the liver expressed transgenic mouse, we didn't see these as many lambda cells and as much escape. And then it later became clear through the work of Tony Baston's lab and others that if a B cell is autoreactive at a later stage of development, it becomes more dependent on BAF, and so the autoreactive cells selectively die when in competition with other B cells. And of course, there must be tolerance at later stages if you if you mutate your receptor to now bind autoantigens. That cell can also be regulated in various ways, probably not by editing, but by apoptosis. So this is sort of our view of how you get rid of autoreactive cells, but of course you never get rid of all of them. There's always some level of autoreactivity that's retained. So now I'm going to switch gears quickly to a project that grew out of this. So we spent a lot of time trying to get editing to work in an in vitro setting using IL-7 cultured B cells, and that did work. But what we were looking for was, we were looking for a master regulator of recombination, which we didn't find, but probably the work of Mark Schlissel came closest to showing that there's transcriptional regulation of RAGs that could, through FOXO1, that could be important in our system. But in the course of these studies, we compared editing cell cultures with cell cultures where the ligand was absent. And what we noticed was that when ligand was absent, the B cells matured slightly. They upregulated markers of maturity, CD22, CD23, and other markers. So we started wondering what all of these genes were that were upregulated and what they were doing, because a lot of them seemed functionally useful and important. And one of the genes that came up, we ended up calling mouse PLD4. And we worked on this as a side project for many years, different people in the lab. And so after many years, we figured out that PLD4 resides here in the endosome, endolysosome, and it has and PLD4 is very sort of tissue specific, but it has a partner or a homologue called PLD3, which does very similar things. And they're both nucleases that live in the endolysosome, and they can degrade single-stranded RNA and DNA, provided they lack the 5' phosphate, which seems strange, but it turns out the endonucleases in the lysosome actually cut, leaving a 5' hydroxyl. And so these enzymes are very efficient at clearing out the fragments of DNA and RNA in the endolysosome. Anyway, so we did a whole series of studies with these. We knocked out the genes and tried to look at the phenotype. If you knock out PLD4, you get an autoinflammatory phenotype, a macrophage activation syndrome, and you see upregulation of MHC class II on peritoneal macrophages, splenomegaly, high levels of interferon gamma in the blood, CXCL10, and what you could call an interferon signature, even in mice that lacked RAG. So this seems to be an innate immune phenotype. And we finally traced most of the phenotype to TLR9. So if you knock out TLR9, you normalize almost everything here. So these mice develop something that looks like macrophage activation syndrome. And interestingly, we independently in Jackson Labs, a mutation was found in the PLD4 gene of BALB/c mice, and those mice get a very distinct looking phenotype. So the mice are runted, they have hair problems. They also have a lupus-like phenotype. They make a very high anti-DNA titer. And those mice also, you can normalize by knocking out TLR9. So it makes me think there's somewhere in a pediatric clinic somewhere, there are human beings with deficiency in PLD4 that have interesting rheumatological phenotypes, but they haven't been described yet. And if you hear of something like that, please let me know. The PLD4 was actually associated by in GWAS studies with systemic lupus. So if you look at these different markers, here's PLD4. This was mainly in an Oriental population. I think this was a Japanese population. But it's also been linked to RA and systemic sclerosis with weaker association. So this is just showing some of the nuclease activity of the enzyme. The PLD3 has been linked to Alzheimer's. So here is what's shown here is the frequency in the population of variants or of missense mutants, whatever you want to call it, with the risk of Alzheimer's. And here you see the very famous ApoE4 associated with Alzheimer's. Here's TREM2, and here's PLD3. At the time when this slide was made, it wasn't known what PLD3 does, but it was shown that PLD3 was associated with late onset Alzheimer's. We crystallized both of these enzymes with the help of Meng Yuan in Ian Wilson's lab. And they have beautiful structure. They look very similar, although they have about 40% nucleic acid identity. And we could show that the single-stranded DNA goes into this groove, and the active site is right here. And so this is an enzyme that nibbles nucleotides one at a time off the 5' end. And I told you before that this enzyme requires an unblocked 5' end. So we thought we'd be clever and co-crystallize the enzyme with a substrate that couldn't be cleaved, so we used 5' phosphorylated oligo. And this will be confusing to you, and it was confusing to us, but when we got the crystal structures, we found the 5' phosphate was gone, and it was attached to a histidine on the active site here. This is PLD4. In the case of PLD3, we actually never saw the rest of the nucleotides. They were simply gone. It looks like we know a lot of details. I won't bore you with this, but it's fun for us to think about. So just to summarize this section very quickly, so these enzymes are 5' exonucleases. We did a lot of studies where we knocked out both genes. The PLD3 mutant has a very subtle phenotype, but when you knock out both, you develop a disease called HLH, which is a very serious, life-threatening disease that sometimes comes up in human patients. This is a spontaneous model of that. And we wondered if the whole disease was driven by nucleic acid sensors, and indeed it was. So if you knock out TLR9, the mice get much better. But if you knock out both TLR9 and STING, they're almost completely rescued. So we don't understand why STING, a cytoplasmic sensor, would be activated in mice that have a defect in the endolysosomes, and that's something we'd like to follow up. And finally, recently, a group has shown that PLD3 and 4 have a third enzymatic activity besides exonuclease and phosphatase, and that is they're a BMP synthase. And BMP is a lipid found pretty much only in lysosomes, and it's not clear what its function is, but there is evidence that PLD3 and 4 can help synthesize this as well. So we'd like to understand what that all means. How much time do I have? I might have to stop here.
No, we have 10 minutes.
Okay. So so let me move on then. Okay, so this is exciting. So this is a postdoc, Yung-Shao Lee, who did an amazing, found an amazing observation in cancer. And very briefly, what she showed was that if you block plasma cell differentiation in a couple different ways, mice actually become more resistant to tumors. This is illustrated here. So she took mice that have a B-cell restricted deficiency in BLIMP1, which is a master regulator of plasma cell differentiation. And so these mice can't make, they have B cells, but they can't make antibodies. And as you can see in the control mice with just the floxed BLIMP1, the tumors grow. This is a colon tumor. It grows at a certain rate in the control. And if the mice can't make plasma cells, the tumor grows much more slowly. And so this was, we did a lot of controls and a lot of repetitions to understand this. The CD19 Cree has nothing to do with it. If you just make a CD19 Cree mouse, tumor grows the same rate. And if you look at an independent mouse that can't make plasma cells, so this is the IgMI mouse. So in that strain, there have been strategic deletions within the immunoglobulin heavy chain locus to knock out downstream isotypes and exons involved in secretion of IgM. And in that mouse, you can make no plasma cells, but you have BLIMP. And again, we saw that B cells or tumors grew less well in the absence of plasma cells. And she also looked at different tumors, B16 and Lewis lung carcinoma. And again, the absence of plasma cells was beneficial in regulating tumor growth. And correlating with this is much better, higher frequency of interferon gamma producing T cells, of granzyme B producing CD8 cells, and this is the CD4 cells expressing interferon gamma as well. We saw higher, in the absence of plasma cell differentiation, we also saw higher effector memory cell frequencies and fewer exhausted CD8 cells. So we then went on to do some single-cell RNA-seq types of experiments. So we looked first at the whole transcriptome and also at VDJ sequences. Sorry about there's something, there was a glitch there. So if you just sort of superimpose control and BLIMP1 knockout B cells from tumor-expressing mice, so these are tumor-infiltrating cells, you see the plasma cells are missing from the BLIMP1 knockout mouse that carries tumors, as expected. And we saw another cluster of cells that seemed to be expanded or present only in the BLIMP1 knockouts. These turned out to be enriched for expanded clones of B cells. And these cells had a phenotype a little bit like a memory cell, but not exactly. They had expanded clones of heavy and light chain genes that a couple of them that we expressed, and I'll show you that in a minute. And there was, and they also carried some markers, unusual markers, CD9 and CD148. So these B cells were, this class of B cells was expanded in number. And so at this point, we concluded that probably precluding plasma cell differentiation didn't enhance tumors because antibodies were counterproductive. It's that we redirected differentiation to a more memory phenotype rather than a plasma cell phenotype. So of course, everyone would like to see translation of something like this to something that you might use for patients. So miraculously, there is actually an approved drug that suppresses BLIMP. And this is VPA valproic acid, which is an approved drug for epilepsy and some other conditions. And it actually suppresses BLIMP, and you can show it suppresses plasma cell formation. If you give this in the drinking water, you get fewer plasma cells. And you suppress tumor growth in a wild-type mouse. So these are wild-type mice that are given this drug. And at the highest concentrations, it's almost as good as knocking out BLIMP1 in suppressing tumor growth. It's like magic. We then, Jung Shao went on to treat a spontaneous tumor model. This is a mammary tumor model that develops in females tumors at a certain age. If you if you put the mice at 6 weeks of age on VPA in the drinking water, their rate of tumor growth is greatly suppressed. So we think this is a model that could be taken advantage of, maybe in conjunction with other therapies, to perhaps suppress tumors. So that's my story. Slightly B-cell related at the end. And I just have a lot of people to thank. I mentioned Kurt Birke, who helped us early on. I had some technicians in Basel and a lot of friends who helped me get together, get started in this field. And then In Basel, in Denver, we studied other aspects of editing, and a lot of these people did some sort of key work to help people believe in the editing model and to study it in more detail. And more recently, this is lab circa 2005 in La Jolla. And more recently, these are the people who contributed a lot to the PLD4 story. So I'll stop here and thank you all again for your generosity.